like last Sunday, we started with the chapter cellular fate processes, where we talked about one cellular fate process, that is differentiation. And we said that process of differentiation can be described in three ways. Like we can measure it as a change in gene expression. Then we can also measure it as a function of change in cellular function, basically how due to differentiation cells function is changing, so we can use that as well. And also we can describe it using mathematical equations. So only this mathematical equation part was left. um, the process of differentiation is described or has been described using two mathematical models. We will look at each model one by one. So as you know, the process of differentiation is very slow, and because it can take weeks, a lot, like large number of weeks to complete, except for the erythropoiesis and etc., which are relatively faster. Other processes can take a long time. The kinetics of complex differentiation process, it can be described mathematically using two models. Okay, one is called compartmental model, another is called continuous model. Now for any, if you are familiar with this kind of studies where people use maths to describe something, so they have they usually take some assumptions in order to make their model much more simplified and uh, in a way that we can write it in form of maths. So they they remove certain real parameters and simplify the entire scenario. So here also, in this particular mathematical model, the compartmental model, and as well as the continuous model, in both this model, they have taken the assumption that they are not using any molecular information. So whatever changes that is going on at the gene level, those changes are not taken into account. And also both models are cell population type models. So you will understand this in a few minutes. So first model is the compartmental model. So the differentiation process, they have divided into stages, or you can say compartments, discrete compartments. And in each compartment, or each compartment is uh, displaying or is represented or representing one stage in differentiation. So you have seen in erythropoiesis, the cell uh, structure and all is changing, right? So there was early precursor cell, and then progenitor cell, and etc. So you can place all the early precursor cells in one stage, okay, and then the later progenitor cells, etc., in another stage. So similarly, this is what they are doing in compartmental model. So if I is the influx of cells that has entered the lineage or has committed themselves to get differentiated into particular cell type, this is the initial number of cells. Then at say stage 1, yeah, 1, this subscript 1 is representing the stage. Okay, so here this is the x1 number of cells are there in stage 1. Then in stage 2 there is x2 number of cells, and similarly in stage I there will be xi number of cells. So usually the number of stages n, here n, it can go up to 16 to 18. So now you can understand why this process is relatively slow, because it is taking place through a large number of stages. And here Ki, or the K1, K2, what are those? Those are called transition rates. That is the rate at which the cell at say stage 1 is undergoing change and then entering another stage 2. So this is the transition rate of cells at stage Xi to stage Xi plus 1 is denoted by Ki. So for each stage there will be K1, then there will be K2, then for stage I, the cells at xi minus 1, they are transitioning at the rate of ki minus 1, and now they have entered the stage ith stage. The fully differentiated cells, they have a finite lifetime. Sorry. They have a finite lifetime, all right, so they can only live for few cell division cycles and then they will die. Okay, so and also death happens at a certain rate, and this process is irreversible. Please note, this is an irreversible process. The conditions like transdifferentiation etc won't happen. That is why in this scheme that you are seeing, there is no reverse arrow. All the arrow is in one direction only. All the cells at a particular stage of differentiation, they are assumed to be identical. Okay, identical in all sense like morphology and then cellular content etc. Everything is identical for all the cells that are present at a particular stage. That is why each stage is called homogeneous, because all the cells are identical. Okay, now I is the flux, means input flux, means how many cells have committed to get differentiated into a particular cell type initially at the time t0, say this is the flux. And then this scheme, like this flow of cells from one stage to another, it can be mathematically written using this expression. This is a first order differential expression where dxi by dt, this is the change in cell number with time, right, is equal to k I minus 1 into x I minus 1. So this is the number of cells that were there in compartment I minus 1. Now these cells, they will move to compartment I. So there the cell number will become ki into xi. Okay. Now this is the change in cell number is happening and is changing with time. So here xi is basically a function of time here. So yes, Vani, I have received your answer sheet. Okay. So as I was saying, x here is the function of time only. You understood why it is function of time? Because with time, the cell number is changing in each compartment, right? Because cells are moving forward from compartment I minus 1 to the next compartment, ith compartment. This is the mathematical equation that is used to describe this dynamic nature of this compartmental model of differentiation. Now, this transition rate from one stage to the next is proportional to the number of cells present at that stage. Means if the cells present at say ith stage is very large, okay, then the rate at which it will move to the next stage I plus 1 will be high. Okay, so this is a proportional thing, and this transition rate also may be the function of uh, cytokine concentration, because cytokines, they are what, they are signaling molecules, right? So in their presence, more and more number of cells will switch on their biochemical pathways inside their cells, and uh, will be expressing only a fixed set of genes, subset of genes, and start to express the characters of to be differentiated cells. So the transition rate here is a function of both the cell number as well as the cytokine concentration. Now if you look at this differential equation, okay, and this is a not unsteady state equation. Okay, if we want to make it steady state, then what you will do, you will take this change, this value will become equal to 0, right? Because at steady state things doesn't change with time. So dxi by dt will become 0. So that will give you the expression for steady state. So at steady state you can derive the values of cell number that is present at each compartment. Can you do it? Do you think you can do it? Yes or no? Anyone present in this meeting?
Yes, ma'am.
Yeah. So I was asking, if I change this expression to a steady state expression, would you be able to find out the number of cells that is present at any particular stage, say IS stage? Would you be able to calculate that from this expression? Yes?
Steady state, everything will be constant, right?
Yeah. So at steady state, this in the right hand side, sorry, in the left hand side, this dxi by dt, this value becomes 0, right? Because the cell number won't change with time. Then would you be able to calculate what will be xi minus 1 or what will be xi? Would you be able to calculate?
No.
Why no?
We can try.
So it's going to become simple equations like this, right?
Ai minus xi minus equals to ki.
It becomes simple equation like this, right? And then you will shift this value to left hand side, right? And then if xi is required, then you can calculate xi. So say at time t equals to 0, when some some cell population have committed themselves to undergo differentiation, then what will be the expression? Dxi by I is an influx, right? That is the number of cells that have committed, minus k1 x1. So if you look at this, so here I is the number of cells that has committed themselves to get differentiated, and then they will move on to the next compartment, that is 1 x1 compartment, correct? So at initially you can write this expression. Now at steady state this expression will change into what? Here this left hand side will become 0 because it's steady state, okay? Minus k1 x1. Now if not, unknown is what? Unknown I, you know, k is some rate that is given to you. Unknown is x1 because you don't know how many cells are going to be there at compartment 1. So then you can calculate x1. Similarly, at some time t1, when the cells have moved to the next compartment, say second compartment, then you have to first frame an equation like this, right? Assume steady state, and then you can calculate the how many cells will be there in compartment 2. Sounds easy, doable.
Yes, ma'am.
Yeah, correct. So in this way you can calculate the cells that are present at any particular compartment, okay? And they are moving, right? This model assumes that as the cell is gaining a differentiated function, it is moving from one stage to another stage. That means it is moving from, say, left hand side when the cell was completely immature, and moving to the last stage where the cell has become mature. So degree of maturation is increasing as the cells are moving from one stage to another stage. Okay. Now what you can do, if you do a cumulative sum, like if you take a summation of the values of all the values, this x1 values, then you will find some equation like this. Okay, summation. If you take a summation of that, you can try it yourself. and this tau diff, that is a diff stands for differentiation, is the overall time constant. That is the time it will take for the entire process to finish. So this comes from the summation of all the values that you obtain for each stage, stage 1, 2, 3, 4, like this. Okay, clear? Is there any doubt?
No, ma'am.
Okay, no doubt. Now we'll move on to the next model that is used for describing the process of differentiation. Now this is a continuous model. Now this model assumes that once the cells have committed to undergo differentiation, the process pretty much proceeds at a fixed rate. So in the previous model, uh, for cells to move from one stage to another, there is some rate associated with it. So the rate is changing and it is dependent on the cell number and the cytokine concentration. Now this model assumes that once the cells have committed to differentiation, the differentiation entire process will move, uh, like there are no compartments. It continuously moving, and that movement of cell population towards the mature state is happening at a fixed rate. So this model is expressed or described using this mathematical expression. Now this is a first order partial derivative equation, okay, where x is the cell number and this cell number is changing with time. So del x by del t plus, now del here is the rate of the differentiation. As I said, it is moving, the differentiation process is proceeding at a fixed rate and this fixed rate is given by del. Okay, and this del is itself a function of another parameter called a. Okay, this parameter is denoted by a. Now this a is actually a measure of maturity of the cell. So it can have a value between 0 to 1. If a is 0 means the cell is very much immature, means it is a stem cell, and when the a value have become 1, that means the cell is completely mature and has gained a particular specialized function. So a is basically a parameter that measures at what state of differentiated condition it is. Okay, so it can have a value between 0 to 1 only, like it can have a point value as well. Understood? So, and x, the cell number x is also a function of a, and this entire equation, this left hand side is equals to 0. This is a partial derivative equation. Now as you know, uh, here x is now a function of two parameters. Now means from this equation I think you can understand that x is a parameter of two things now, both time and a. Means the cell number is changing with time, correct, and the number of cells that are present at say particular differentiated state is also changing. So it's a function of t as well as a. The cell number is a function of both t and a. Whereas in the compartmental model, x was only a function of time. So with time, the number of cells are changing as it is moving from one compartment to another, and also the movement is happening at different rates. Whereas in the continuous model, the movement is happening at the fixed rate, okay, and the cell number is changing with time, as well as uh, at a particular differentiated state, say a is equals to 0.1, say a is equals to 0.1, the cell number will be different. So in this way, you can understand how the differentiation process is described using maths, using these two mathematical models. Okay, is there any doubt in understanding any of these two mathematical models?
No, ma'am.
No. So what about others? Uh, Shambhavi, Vaishnavi, are you guys there? This teams dot meet. Yeah. So is there any doubt in understanding so far?
Actually, I was facing network issue. I'll again watch this video.
Okay, okay, fine, fine. So if you find any difficulty in understanding later on, then let me know. Okay.
Yes, ma'am.
So this was all about the differentiation process. Now we will move on to the next cellular fate processes, that is cell migration. So from this term you are able to understand, here I am talking about the mobility of the cell. The cell is moving from one place to another. So it's a very important function, basically means it's a very important cellular fate process, because it is important for many functions of the tissue as well as for some pathological processes to happen. So if cell migration doesn't happen, then several physiological processes won't be able to proceed, as well as some pathological processes like cancer, etc., take help of this cellular fate processes in order to continue. A cell migration is important during organogenesis, means during formation of an organ, as well as during embryonic development. You guys have seen during embryogenesis how the layers are formed. So during that stages, the cell is moving actually, correct? It is dividing and then moving in order to create unique structures like gastrula structures that I've talked about, tube within tube structures. So those were all possible because the cell was able to migrate. Then cell migration is also important for the tissue repair process. Again, you are familiar with this. Here, a large number of cell populations migrate, like inflammatory cells and then tissue cells, etc. Then blood vessel cells. All these cells, they have to migrate and then heal the area. So there also cell migration is important. Immune system relies on cell migration, correct? The blood cells, they are migrating throughout their body all the time, as well as some pathological conditions like cancer metastasis, where the cancer spreads throughout the body. So how the cancer spreading is happening? It is happening because the cancerous cells is migrating, correct? They're migrating from their source location to some other target in the body and thus spreading the cancer. And sometimes you can actually uh, trigger cell migration, okay, of stem cells as well. Stem cells can also migrate from their native location. So people have injected some chemotactic growth factors. So these are basically the signaling factors that is going to uh, signal the cells to come and migrate towards the source. So these are chemotactic growth factors. We will talk more about the growth factors in the next chapter, but I think you are able to understand what I'm talking about here. So when the patient is infused some these kind of factors, growth factors, then people have seen that they are able to induce cell migration, induce the migration of the stem cells from the bone marrow, and they come out and started circulating in the blood circulation. Because what happens is cell migration always happens towards the high concentration of growth factor. So if this is the cell, okay, and here it few molecules of growth factors have arrived and got attached to some cell membrane receptors. So here the concentration is low of the growth factor, and it will try and move to the point where the growth factor concentration is very high, because to the cell, if I'm talking about in perspective of a cell, it feels that it is being called, मुझे बुलावा आया है वहाँ आने का. So in this way the migration happens, okay. A concentration gradient it has to sense in order to migrate. That is what I'm talking about. Now cell migration is like, as I said, it happens in presence of concentration gradient. So this is basically a type of a stimuli. You are triggering it to migrate, you're inducing it to migrate. Now the growth factors are not only means not only using growth factors you can do cell migration. There are several other kinds of stimuli as well through which you can induce cell migration. So first is chemotaxis. First mode of cell migration is called chemotaxis, where you use growth factors. Now growth factors are also called soluble factors because they are very small and get easily solubilized in water-based solvents like blood, etc. So that's why they are called soluble factors as well. So if a cell senses the presence of a concentration gradient of a growth factor, then cell migrates towards the source, to where the concentration is the highest. Also, if there is a growth concentration gradient of insoluble factors. Now, insoluble factors means ECM usually. Okay, ECM components, because ECM components, as I also already mentioned, these proteins or polysaccharides, etc., these are very large molecules. They cannot migrate. Their diffusivities are very low. Okay, because of their large molecular weight. So they are insoluble in nature. If a concentration gradient is established of this ECM, then also you can trigger cell migration. And this type of cell migration is called haptotaxis. Then in presence of electrical currents, cell migration happen. This is called galvanotaxis. Now, if you change the surface topography means surface roughness or smoothness, texture of the surface. So based on how you are designing the surface texture, you can influence the cell migration process. And this type of migration process is called contact guidance. So people have patterned surfaces and triggered or induced cell migration in such a way that they are moving in only one line only, but in the other line they are not moving. So we will discuss a paper related to this later on. Okay, and another mode of cell migration is called contact inhibition. So you guys must be familiar with this. Here what happens when the lamellopodium means the extensions of the cell membrane. So cell is not completely spherical, it has this kind of structures, finger like structures. These are called lamellopodia in plural, and singular is called lamellopodium. So they they extend, they try to spread. That is called cell spreading. That is the extension of the lamellopodium is called cell spreading. So when two adjacent cells, the lamellopodium, when they meet, then the cell migration stops. This is this mode of cell migration is called contact attenuation. So these 5 types of cell migration is known to us that people have used to their benefit. So in the next class, we will talk about in detail like how a cell migrates. So we will talk about it in the next class. As I'm seeing, time is already close to our end. So is there any queries? Then let me know. Anything that you have not understood?
Ma'am,
Yeah.
how will we study these haptotaxis and all, ma'am? Because it happens in a very micro level, right? What instruments do we use, ma'am?
What instruments do we use to measure this?
Steady or even measure, ma'am.
I didn't get your question. Can you reframe it?
Yes, ma'am. These are very miniature level happenings, right?
Yes,
So,
micro level.
what
Yeah.
kind of experiments we use or study we do to understand how it happens?
So we will talk about it. We'll talk about the experiments that people use to measure cell migration and quantify it using some mathematical expressions. We'll talk about it. Okay.
Okay, ma'am.
Any other queries?
Ma'am, actually, there is a request
Yeah. Yeah.
uh, to extend the duration of test, right?
Yeah.
Ma'am, please extend the duration of test because it's miscellaneous question and it is, you know,
Uh, you want less quizzes to happen? Is this
No,
what you're?
ma'am. No, ma'am. I was just saying that please extend the duration. Means it instead of 15 minutes, it should be of 20 to 30 minutes, so that it would be.
Easy for you. Okay. So was that was today's paper a bit difficult?
Ma'am, not
Not all.
difficult, but we take time to like read and then processing in our mind.
Okay. So you want paper to be like a bit more time, like 15 minutes, say 30 minutes, या फिर 25 minutes ऐसा कुछ. You're asking that, right?
Yes, ma'am.
Okay.
Because it's almost 30% of time is gone in thinking whether to answer in one word or in a paragraphic form. So
Okay, okay, fine, fine. I understand that. Okay, I'll from the next quiz onwards, I'll keep the time bit longer so that you guys can finish it in like within time and you can write the answers properly. Okay, I'll give you more
okay, ma'am.
time. Okay, from next
Okay,
quiz. Okay.
Thank you, ma'am.
Yeah, I'll
thank you.
yeah. So let me stop the recording and then I'll take attendance.